Many thanks for the introduction. Yeah, my name is Marcin Wierzbiński and I am a scientific programmer at Sano. And today, welcome everyone to today's seminar. In this presentation, I will present the results from the work on the spatial inference of cell type decomposition. Okay, one question, the important thing is that this presentation is with collaboration with Agnieszka Geras, which is the author of Celloscope. I will present more. Okay, so the challenge for scientists from bioinformatics is determining the spatial arrangement of different cell types across the tissue. So to address this problem is useful the technique, which is spatial transcriptomic technique. So the spatial transcriptomic is a molecular profiling method that allows measure all gene activity across the tissue and map where the activity is occupying. So all information is organized as a spatial transcriptomic microarrays, which contain a grid with the localization with unknown number of cells and the nature of the spot measurements. So we can observe the signals which comes from mixtures of cell types decomposition. With each spot localization, we have a gene expression with a count number. So this is the integer number. So, and this data is represented as a matrix. So we can observe that on the bottom panel. And on the up panel, we can observe the ST microarrays, which means that we can observe the tendency of our growing within tumor. So this example, in this place, we can observe the prostate cancer tissue. So here we can observe the area and the spatial aspect of this tissue. So the relationship between cells and localization within tissue is important and critical to understanding normal development and disease pathology. So we can address the problems to solve. So, for instance, the first of the problem is to build a model that identifies the cells type decomposition. Of course, based on the spatial transcriptomic data. The second is to identify the proportion of types within spots. So based on the knowledge from the spatial transcriptomic, we can observe the spots. So this is the places, the places when we have a number of cells in these places. So the spots contain an unknown number of cells. So it means that we don't know exactly the number, how is big and so on. So, and we don't know also the exact decomposition of the types for those cells. What is more, we want to model the spatial relationship between the proportion of those types. So it means that our tumor can grow like spatial area, like going to direction, but with spatial dependencies. So that I think it's nice to show this problem as some machine learning problems. Building a model is similar to some analogy from machine learning. So that this is called unsupervised learning, which is a type of algorithm that learns pattern from untangled data. So it means that we have some input and we want to infer about the data with specific restriction. So in this place, we have a model input. So a priori knowledge about our spatial transcriptomic localization and marker gene expression. So it means that we have the information for each gene, how is the level of this specific gene as a number. So we have no information about the decomposition. So it means that our output of model, we don't have the exact information about the decomposition. And the one possible method to solve this problem is a probabilistic graphical model. So which means that we have a certain graph structure that can encode the dependency between our random variables. And I will discuss in this presentation about how we can express those problems in a probabilistic graphical model. Okay, so here Agnieszka Geras with authors propose the Celloscope, which is a Bayesian probabilistic graphical model of gene expression in spatial transcriptomic data. So this model deconvolutes cell type composition in spots, and this model has a method to infer parameters based on the Monte Carlo Markov chain methods. So it means that we have a collection of random variables, and we can infer about the distribution from this collection. So the Monte Carlo Markov chain method means in this place like this. So that our data, we can assume that, for instance, in this panel, we have a variable CGS, which means the expression level of a gene G in a spot S, and this variable is modeled using the discrete probabilistic distribution, the negative binomial distribution. And this distribution has two parameters, R and P, and these parameters will be the variables from our probabilistic graphical model. The number of cells in this model is estimated based on the images from spatial transcriptomic, and prior knowledge on a marker gene. Marker gene means that our gene is highly expressed. For consider cell type is encoded as a binary matrix when we have the indicator that our gene is marker or not. Very important thing is that this Celloscope model assumes that the measure expression depends on a hidden cell type mixture in each spot from our ST microarrays. Okay, here we have a histogram of with density for a fixed gene, LAMP5, counted for all spots, and we can observe that this this histogram shows that our distribution is very similar to negative distribution for this gene and our counts. So with those parameters, mu equal to 3.98, sorry, and k equal to this guy. Okay, so let's see this model as a structure graph. So this model, the Celloscope, estimates cell counts and the binary matrix based on the a priori knowledge from the gene expression. So here we have input for this model. So the input means that this model get this two matrices and infer about the hidden variables, and the output is a cell deconvolution for within specific spots. Of course, the variables is quite complicated because we want to, for instance, infer about the present, the specific type within this spot, this spot, for instance, because sometimes not all types are in the specific spot. So the model from the assumption from previous also slide, we have this model assumes that the hidden cell type mixture depends on our input. So here we have a first variable, the binary variable from Celloscope model. So here is a variable. We know exactly the information based on our ST data, and this variable means the indicator whatever gene G is marker or for type T. Okay, and here we have the two values for those information. And so from this side, it's important to indicate the high expression, so that our binary variable is useful to set the higher average expression overexpression level of a marker gene G in the specific type. And we can see the information about the sum between average expression level shared across all genes for types T and the specific, and also the overexpression level of a marker gene G in its specific type T. So this equation is higher when our gene is marker gene, because B is equal to 1, so it means that our expression is higher. Okay, so that is the assumption from this model. Okay, so the quite useful thing is to model the gene expression level. So we want to prepare this good assumption for the distribution for expression level, and we know only that our expression level is a discrete variable. So it's nice to use the discrete distribution from probabilistic, and also we can observe the tendency to be as a negative binomial from our previous histogram also. Okay, so and the negative binomial distribution has two parameters that are modeled in Celloscope as a hidden variables, of course. Recall the CGS is expression level of a gene G in a spot S. So it means that we have two very important information, that our gene and also the localization for this gene on the ST microarrays. So the assumption of average gene expression level of a specific gene G for that cell is equal to the to this guy from the bottom panel. So, and this sum is very important in this model, because this is the information about how we can, for instance, sample from negative binomial for preparing the simulation data, for instance, or something like also the information about how we can observe the mean level, the mean expression level for the for those places. Okay. So here is HTS means the hidden hidden proportion for type T within the spot S. And this guy is expressed as, of course, from previous slide here, the expected average expression level for a single cell. This is the place when we use the properties from a negative binomial, because negative binomial has a useful property with the two independent variables from negative binomial. So that it's in this place we can express the information that the sum is equal to the sum of the independent negative binomial variables is equal to this distribution. So, and this equalities means the distribution equalities. Okay, and here we have the information. The NS means the number of all cells in spot S. And here is a hidden decomposition for type T within this spot. Okay, so that is a priori assumption for our input data. So we have two input data for our model: the expression level CGEs and beta and BGS, which means the information about our gene is marker or not. Okay. Yeah. So that this slide represents the graphical representation. So because our graphical probabilistic model can we express as a graph with dependency between variables, so the directed graph and information. So for instance, the information about the variables, it's for instance, gray nodes correspond to the observed variables. Double circle represent the deterministic one. So it means that this variable is computed using the variables from previous arrays, and the remaining nodes correspond to hidden variables. So the latent variables. It's a similar explanation. Arrows represent probabilistic dependencies that we can encode the conditional distribution and conditional dependencies. For instance, this variable mu GEs depends on the few variables from this side, and we can observe this dependency from this guy. Yeah. So the HTS means the hidden fraction of type T in spot S. here is a base expression for the types other than specific shard across all genes. And beta GT means the indicator as from previous slides. Okay, so the deterministic variables means that our number, for instance, can be computed using the previous variables. Here, so it's an important explanation because the HTS within the spot should sum up to 1. For a specific spot, when we have the range of types, so we need to sum up to 1. So that this is the form for that. Okay, and here we have a conditional distribution for information. For instance, we have the indicator that our type is present in a specific spot or not, because sometimes, of course, some spot doesn't contain the type, some type, for instance. So that also so it means that we want to assume that here we have a gamma distribution, which is a continuous distribution, and this gamma has 2 parameters, and the parameters we can observe on this graph, which means that when our parameters is set for a specific parameter, that this information, when we sample from this distribution, has a huge number, for instance. So it means that our type, the fraction should be large, because we have the 1, so it means that this type is presented in this spot. Okay, so that is important. We have the 2 types of variables: observed variables and latent variables, the hidden variables or latent. Okay, and this is the representation, and it's extremely useful because from probabilistic, it's sometimes we have the problem with model the huge space of variables. Yeah, so that this representation can be useful to build a inference method, for instance. Okay, and here we have details about the parameters inference. So it means that, of course, we have 2 types of variables: the observed variables and the latent variables. Okay, and in this model, Celloscope used the metropolis within Gibbs sampler to infer those variables. Okay, sometimes some variables from this, for instance, lambda 0, is set as it means that for instance, from this part is set as a single number. Yeah, so it means that we don't have the random variable, we have the number like as a parameter, but it's not often approach that. So for instance, CG is a variable, the variable, BG is also, PST also, and so on. So it means that we have the structure we want to sample from the conditional probability, like here. for instance, and here. So it means that we only assume about this distribution, and we want to sample the PST from this combinational distribution, like here. Yeah, so that our distribution is up to some constant. And we want to sample from this product for a priori distribution and proposal distribution. So, and it's extremely useful to use the two methods with specific linked between the Gibbs sampler and metropolis hasting. So it means, for instance, in Gibbs sampler, so this is the algorithm, the probabilistic algorithm to sample from conditional distribution for a specific step. And the Gibbs sampler produced the hidden Markov random fields. So it means that this sequence of random variables goes to the desired distribution in this setup. So, and the metropolis hasting is useful to find the formula to express the combinational distribution when we have only the information between like product a priori distribution and a proposal distribution without the normalization constant here. So that it's important to say that this information is useful. This two methods is useful to infer from our model. Okay, and here is a slide from a publication from Agnieszka Gera's publication with authors about the results obtained for simulated data. So the result, the simulation study demonstrated the Celloscope accurate performance. So it means that it's a very accurate method. But one thing to say is important, because our simulated data based on our assumption from this section, yeah. So it means that it's, I think, only useful to say that our model can work, not like works, because in this place we have an assumption to with specific thing, like I assume that our expression gene level has a negative binomial and so on, yeah. But from nature, it's a quite problematic, yeah, because we don't have the exactly information how we can decomposition within the spots, for instance, yeah. So it means that this results, it's very accurate, yeah, but only for the simulated data. So it means that we just assume we just sample from negative binomial and compute the mean expression based on this information, and also we sampled the marker gene array and some information from that, yeah. So, but of course, the Celloscope was compared with 4 methods like cell assign, RCTD, and stereoscope, and the 2 information. When our cell counts is known, and also the cell count we have as a priori. So it means that we just sample from the distribution for this variable. Okay, and the average error across a spot is equal to 0.01. So it means that it's super slow. Yeah. But for instance, another methods also has a nice average, and we have two scenarios. This sparse scenario, so it means that decreased number of types per spot. So it means that that our numbers is quite low and dense. So it means that our increased number of types per spot. Okay, and here we have another measurement, another information that a fraction of correctly indicated dominant types. So that Celloscope also has the number like going to 1. So it's super information that our fraction is correctly. And this is the bar plot. So it means that we run so it means that the Celloscope was running for the 15 models sample methods, sample data. Yeah, so that is we can observe in this place the bar of error. Yeah, so it means that the median of this is this place, and the distribution of this error. Yeah, so and from this side we can observe that distribution is very high. So it means that it's accurate. It's a very accurate performance. Okay, and here we have a real data set. So this is the mouse brain data set from this website, and it was tested on this 200 at approximately 200 number of spots. Okay, and this is the image from this data set represent for instance the fragment of a mouse brain. Okay, and here what is nice in this data set that we don't have exactly the information about the decomposition within the types, but we have the biological information that exactly the cell types. Yeah, the localization like from these cell types, but the information that how much we have cell types within this micro the ST micro array. Okay, and we can observe that that if we have a few cell types with specific information. Yeah, for instance, the information about what is the type of the neuron or something like, or some another information about this cell and so on. So, and the model has a heat map for represent the spatial composition of a spot. And we can observe that the composition is more or less located within these places. And Pearson correlation means the information about the linear correlation between two variables. So, and we can observe the correlation between specific types. For instance, we have a high correlation between this type and this type. Yeah, and we can observe that this place and this place has a similar colors, like, okay, maybe not colors, but like the shape. Yeah, here and here we have the same shape, the same shape, and also, for instance, here and here. Yes. Okay, and this results shows that model the Celloscope's capability to unravel spots, percentage composition with respect to the cell types, so that the decomposition is useful and it's quite okay. It's okay, sorry. Okay, so here I will propose the extension for the Celloscope. So the name of this modification is a Spatial, because in this model I want to take into the account spatial aspects, which is modeled by this Boltzmann distribution. The Boltzmann distribution is useful in the statistic physics, and this distribution has the property which means the hidden Markov random fields. So it means that we can model the dependency, the spatial dependency between discrete or continuous variables. So the hidden Markov random fields is a generalization of hidden Markov model. Instead of having the Markov chain, we have Markov fields with specific properties, like Markov properties. So it's very useful because our collection of variables has the property, the collection has a useful property to model the sequence data. So from this point, it's important to indicate that I simplified the assumption about the decomposition of a gene expression level. Instead of a negative binomial, I use a Poisson distribution, which is also a discrete distribution from this place. I also remove the variables for to indicate the marker gene. So it means that our model has only need two information. And the first information is the information about the lattice, the lattice structure. So it means that we have a spots. The points are spots, and then this model takes into the account this spot lattice as an input, and also marker gene matrix with information about the expression for specific gene within these places, and infer the information about the type decomposition within those spots but in this place that can map us the information about the color representation, like here. So it means that the composition is we can observe the dependency between the neighborhoods from this place. Okay, and here is a graphical representation of the Celloscope model. The green nodes, CGEs, represent as previous the gene expression level, and other variables represent hidden variables or latent variables. Okay, and here we have the collection of variables in two types: the observable variables and latent variables. And model Celloscope simplify variables of Celloscope, leaving variables as such as unnormalized, abundance of type T in spot S. So, and I changed the assumption for this distribution, because from previous information we had a gamma distribution, and here we have a truncate gamma. So it's extremely useful to model the information from the from the boundary support of the random variable. Okay, and HTS, it's also the hidden fraction of type T in S spot S. And lambda 0, of course, and lambda GT, and so on. So that my now average gene expression in a spot S per one cells can be represented as this expression. And this average gene can be useful because we have a Poisson model with this parameter. Okay, and NS is also the number of all cells in a spot S. But in my model, doesn't take into account the information the estimation of NS. I assume that NS is has a specific distribution, and that's everything. Okay, and my modification, it's important to see this big circle, because we have the specific function f, which transform our random variables theta st into alpha st. So that we have the two important one important mapping between theta st and alpha st. So, and the truncate gamma, it's extremely useful in this sense, because we have the boundary support of this variable. So we just transform this variables into the another one, but this variable has also the boundary support. Okay, and here it's a slide about the assumption for spatial dependencies. So now we have into the power of 2 the dimensional spot lattice. So it's a specific graph of dependency between spots. And each lattice site, so it means each spot, there is a 2-dimensional unit length vector. That, of course, the parameterization of this vector can be represented as a cosine of theta st and sine of theta st. So, and what is important, the intuition behind this model means that our theta st is, okay, sorry for that. So the theta st means the alpha st means the angle for our variable. So that we can think as the angle of a spin between two vectors, and we want to parameterize these vectors as a follow. So it's useful because our vector, we want to flip the vector to have the similarities between two vectors. Here, for instance, when we have the perpendicular two vectors, we want to modify those vectors as we want to find the vectors like quite with less with lower angle between those vectors. Okay, so that's everything from this information, that we have a two-dimensional unit length vector for each spot or lattice site. So this is the similar in this model. So, and the transformation, the one-to-one function that can transform the support of theta st. The support theta st belongs so that we draw the theta st from the from truncate gamma distribution on this interval, and we just map to theta st into this interval, so minus p and p. So this is the one-to-one mapping. So it means that when we have 2 points, that after our mapping we have also 2 points. So of course that we can express this mapping as a equation. Yeah? So that's everything. And the definition of a spin configuration. So this is an assignment of the angle between our spins, and the angle is between minus p and pi. So that's its import. And when we go to the next slide, this is the information about the Boltzmann distribution. I mentioned that the Boltzmann distribution is from the statistic physics. Yeah. So this is the probability distribution that gives the probability that a system, for instance, in our system is a lattice with angles, will be in the certain state as a function that energy, the state energy and the temperature of this system. So, and we can observe that we have an expression for our energy from our energy system. So this is the sum over the whole all lattice site, and our inverse parameter. So the inverse parameter means the temperature of our system. So it means that if beta is equal to 0, our system is totally random. So it means that our spins are independent. When beta goes to it's larger than 0, that our system has a strongest dependency between spots. And the distribution is expressed as here. So the PDF is expressed as follow, when Z means the normalizing constant. So it means that that we can sample from this Boltzmann distribution, but the normalizing constant is very often very difficult to compute because it's this constant depends on the whole space of our spins. So, and I added the Boltzmann distribution to a Celloscope model. So the alpha ST has a Boltzmann distribution. Okay, and here we have results from a toy example. So the data were generated using squares that can move in this place. Each square is it's useful to model a height number of a proportion for the specific type in this square. For instance, yellow colors represent the 0.8 or 9 proportion these types within this area, the square. Okay, and in this place we have only 40 spots, 40 times 40 spots. So that's it's quite short lattice, but it's okay to check the performance. Okay, and here we have the output of the Celloscope model, and on the right side we have the synthetic data. So it means that I have prepared this data for checking the performance. Okay, and we can observe the choral relationship between the composition for types. Okay, so it means that our squares can have information from this can infer the information from the synthetic data, the Celloscope model. The number of iteration for the Metropolis Hastings within Gibbs sampler is set to 10 into the power of 4. So it's quite short trajectory of the hidden Markov random fields. And also the model, this model has a GPU modification, so it means that this model can be run on the GPU. It's extremely useful because the sampling, for instance, is faster 3 times versus the CPU. Okay, so that's everything. In summary, so the Celloscope can successfully locate known types based on marker genes. Moreover, Celloscope is able to indicate spots with potential novel or unknown types. And I propose the extension of Celloscope with name Space Celloscope. This extension can average clusters of cells spatially, and it can be useful to infer spatial clusters of cells and checking the location for those clusters. Thank you for your attention, and please ask any question.
Thank you very much for your quite interesting, very interesting talk, in the sense that it was a good presentation that portion of, let's say, understanding of mathematics, and especially all of these probabilistic phenomena leads to interesting results. So now we have time for question and for discussion. Well, maybe waiting for, oh yes, we have something on the chat. Alex,
Thank you.
Would like to tell you that it was a nice talk. Well, could you please go to this slide, which is on which you have probabilistic graphical model?
Okay, here.
Because I think that it is key to understanding everything. So, well, for example, to which extent such an approach with usage of directed graphs help you in, let's say, solving this problem? What do you, how you make use of this knowledge from graph theory here?
Okay, yeah, it's super useful because, for instance, of course, my talk based on the very short in the information, but in this model, the authors use for instance, the Markov blanket, which is a specific conditional properties from based on the graph structure. For instance, for this variables, yeah, we can check only the childs and parents. So that's it from graph theory, very useful definition. So what means the child and a parent from node, and also graphical probabilistic models. It's they have a few sometimes names, like, okay, it's not a similar name, but it's a quite popular Bayesian networks. So this is the one of the case from probabilistic graphical models. And the Bayesian network also has from graph theory specific properties to compute the conditional dependencies between variables.
Well, I think that we have Ahmed with which follows this direction. So, Ahmed, can you ask this question directly, or you would like to?
Okay, so the question is.
The question is. Have you checked your methods against the graph theoretical framework? So I think that it was, to some extent, you have explained how graph theoretical framework was used, but probably you can add something here.
I can definitely ask Marian. Thank you so much for this very interesting presentation. But as you can see, you're developing a really neat framework. But we are a computational medicine center, and we strive to instantiate all these mathematical and computational frameworks to solve medical problems. So one of the reason why I'm asking this specific question, one of the problems that I'm actually trying to solve, part of the project that we're publishing on right now, is drug repurposing. So we're actually collecting features from the biomedical literature and from clinical trials, and we have networks who can actually entirely transform into the graph model that you're working on. But we're also in the business of identifying like cliques as a smaller communities to pinpoint into possibilities of combination therapy. I apologize, combination therapy for drug treatment. So my point is, if I give you a network coming from literature, and as a matter of fact, I was working on something on the weekend, and I was really hoping to share on Slack, hoping that somebody would jump on it. So I have a very basic model, bare bone model of drugs and disease and genes and proteins and chemical entities. And this model came strictly from the literature, without the probabilistic part, which can actually be gathered if we want to go that way. Now I am really hoping with this model, I would like to see that the combination that we identified from the drug repurposing algorithm of the previous paper can actually pinpoint us with your model into an interesting link that comes and says, well, this clique here is linked to a node that could actually potentially cause a drug interaction or a negative drug interaction. So the direction is very important, and also the positive or negative, the positivity or negativity part of the relationship is also very important as well. Can we use your model to actually transform the network that I'm working on to do that? Is that something that we can maybe briefly discuss here but take offline?
Exactly. I think it's super to discuss maybe offline, but
okay,
I think it's what is important maybe to check the potential assumption of the model, because that our observed variable has a priori assumption of the distribution. And maybe I assume that the problems from drug target identification of drug interaction can have a different assumption for the models.
As a matter of fact, you can actually consider the data that I'm collecting from the literature to be your a priori knowledge, because this is truly the definition of the a priori knowledge. And with your model, you can work with your likelihood functions, and then you can multiply by the posterior probabilities that you're working on, and here you go. You have your Bayesian mantra.
Okay. Yes. Thanks. So that's. I will see on the Slack maybe the potential connection between this model and the drug target identification. Or
okay, I will post something on the general just for the heck of it, and we'll actually have a little bit of attraction, and then see what kind of traffic we get. But I will post something.
Thank you. Many thanks.
Thank you.
Well, I think that this talk at this seminar, it was a kind of general overview of the work which was done in the framework of this, as I understand, Master of Science thesis. But I think that it would be good to look let's say, at this type of models and usage of this type of, let's say, mathematical tools for modeling of other stuff in Sano. And maybe we will ask Marcin to give a kind of, let's say, internal lecture on on something like this, because well, so many details about which somebody could ask question. For example, well, you said at one point that well, we will use gamma distribution. So question is, why exactly gamma distribution?
Yes.
And probably it is there are more of such items which probably require a more detailed
exactly.
explanation. And so I think that we well, let's look at other questions. And do we have something more on chat? No, I don't see any other questions.
Okay, so I haven't checked this method against graphical theoretical frameworks. So because, thank you, many thanks. Because my model, the Celloscope is a modification of Celloscope. It's totally proof of concept now. So it means that I checked I solved the problem how we can model the spatial dependency on a spatial micro array. Yeah, so it's my proposal to check. So it's not now it's not how to say I will check the potential checking on the real data like mouse brain or prostate cancer to check the error and so on.